Hey, hello, everybody. Sorry for the slight delay in getting started today. Because we're slightly delayed, I'm just going to go straight to introducing our fabulous speaker, and then I'll just hand over to her. She's told me, well, both her and I have to run at 3 p.m. our time in the UK, so we might have to cut the Q&A slightly short. Hopefully not, but we'll see. So without further ado, welcome, Camille. So Camille Jeunet-Kelway received her PhD in cognitive sciences in 2016 at the University of Bordeaux in France. After she did, sorry, I'm getting weird proposals. After a postdoctoral fellowship in INRIA, France, and EPFL in Switzerland, she was recruited as a tenured CNRS research scientist in 2018. In 2021, she joined the Institute for Cognitive and Integrative Neurosciences, INSEA, in Bordeaux, where she leads interdisciplinary research on the use of EEG-based BCIs to improve or restore cognitive and motor abilities, both for clinical and non-clinical populations, which I think she's going to cover both today. So really excited. She's particularly interested in studying the learning mechanisms underlying neurofeedback training, as well as the acceptability of neurofeedback procedures and BCI technologies. She has received 3 PhD awards, the European label, and 5 national fundings from the French Research Agency for her research. In 2022, she was also awarded the Early Career Award in Neuroscience from the prestigious BCI Society. And the title of her talk today is already on the screen. So BCI-based neurofeedback training procedures to restore or improve motor skills, a user-centered approach. So very welcome, Camille. Thank you for accepting our invitation.
Thank you. Thank you very much. So can you still hear me well? Perfect. Yes. Okay, good afternoon, everybody. Thank you for being here. Thank you for the invitation. So as Anna said, today I will talk, let's say, for 35 minutes about BCI-based neurofeedback training procedures to restore or improve motor skills, both in clinical and non-clinical applications. And I would like to give a particular stress on the importance of adopting a user-centered approach to reach efficiency and meet patients' needs and expectations. These expectations have been raising a lot in the last years, as BCIs are basically everywhere in the media. And this graph from Gartner that you may know, which is called the hype cycle, depicts the level of expectations as function of time. And you can see that in this most recent version of the Gartner graph, or Gartner cycle, you can see that BCI are here. So they are on the hype phase, and basically after this hype, it says that in 5 to 10 years we will reach a plateau there, and after this plateau there will be a disillusion because expectations would have been too high, and this is quite inevitable based on this theory. But what is important is this part here, where there are 2 ways out. So either the technology is ready and enables fulfilling the most sensible expectations, let's say, or it is not ready, and then there is a risk of entering what is called here the valley of oblivion. And so our responsibility as BCI researchers, I guess, is to do our best to be ready at that time so that BCIs can still continue to be used. So BCIs, and particularly active BCIs, can be schematized as a 4-step closed loop. So I know everybody here knows that, but it's just as a reminder so that we're sure we have the same vocabulary, same basis. So end users, they self-regulate target neuromarkers. So for instance, performing motor imagery tasks to modulate sensorimotor rhythms, while their brain activity is recorded and sent to a machine, the objective of which is to decode in real time. So it means the objective is to identify and extract the target neuromarkers so that information on their modulation can be fed back to end users who will use it to optimize their self-regulation strategy and maximize performance. Interestingly, most current investments in the fields, research and by companies, and therefore most newspaper articles focus primarily on step 2, so on the hardware used to record data, and quite a lot other focuses on signal processing and more generally on AI algorithms used to decode the signals. However, the human in the loop, so steps 1 and 4, are mostly neglected, if not completely erased. And I like illustrating this with a striking example. So sorry, it's written in French, but it means Neuralink is starting a testing phase to determine whether its cerebral implant can control a robotic arm. So here I hope that we all agree that the cerebral implant by itself does not control anything. It's only meant to be a tool used to measure brain activity modulations that have been voluntarily generated by end users and to enable their decoding by specific algorithms. So what I mean here is that we are very much techno-centered, or the media maybe is very much techno-centered in the way they present BCIs. So of course, developing innovative sensors, be they invasive or not, is essential for BCIs to be efficient. But what I mean here is that it's not sufficient, because upstream of this measurement, you need a proper signal. And so it's necessary that end users learn to self-regulate the target neuromarkers. And downstream of the measure, you need a reliable interpretation of the signals measured. So the machine also has to learn to decode end users' commands. So in short, there is what can be called a double learning process that is at play, and it has to be optimized together with the sensors for BCI to reach their full potential. And this optimization calls for a fine understanding of cognitive and neurophysiological mechanisms involved, and thus for a user-centered approach. Oh, sorry, I forgot to say that. Unfortunately, if we look at this graph, it's not exactly this user-centered approach that is used in majority in our field. So this graph shows that you can see here the number of papers published depending on the year, and in blue is all BCI papers, and in yellow it's BCI papers that mention either user learning, training, or any user-related aspects. So you can see that it only represents a very small proportion. And this technocentricity is also reflected in a review that we recently received, and I would like to share with you, which says that at present, the biggest obstacle to the application of BCIs and neurofeedback is still the technical problems, effectiveness, portability, persistence, etc. Therefore, I don't think that this study, which was on acceptability, will make a substantial impact on the field. So again, I insist on the fact that we indeed have a huge technical challenges ahead of us. So this reviewer is partially right. But to me, the causality link, so this therefore is not relevant here, because first, effectiveness may not be enough for BCIs to emerge. Second, because there can be work on acceptability and other user-centered aspects in parallel of the work done on portability, persistence, etc. And third, maybe most importantly, because improving the learnability, the usability, and the acceptability could certainly result in improving the effectiveness of BCIs. And the objective of the next part of my talk here, following the idea and principles introduced by Andrea Kubler and colleagues already 10 years ago, is to convince you that a user-centered approach could enable a drastic improvement of BCI usability and acceptability, and also of their effectiveness. So this I will illustrate with using studies that we have led recently in our team on EEG BCI-based neurofeedback training procedures targeting sensorimotor rhythms in the objective of restoring or improving motor abilities. So first, as a quick reminder, sensorimotor rhythms, SMRs, they are oscillatory activity in the 12-15 Hertz band, according to the neuroscience literature, but usually in BCI literature, it encompasses a larger band, including mu and beta bands, so 830 Hertz. SMRs can be measured using the EEG over the sensorimotor cortex, but there are also beta rhythms are also present in subcortical structure like the subthalamic nucleus, and there are cortical, subcortical networks involved. And what is important here as well is that variations in dopamine levels correlate with modulations of sensorimotor rhythms and have a major impact on motricity. So I will come back to that later when we will talk about Parkinson's disease. So in short, we know a lot about sensorimotor rhythms, but there are still some mysteries, and notably an egg and chicken problem, maybe the bare understanding of the origins of beta activity and also of its precise functional role. So to investigate those mechanisms, we can, for instance, use motor imagery, which consists in imagining performing a movement without actually moving. So this task of motor imagery, it's most of you already know, but it's very interesting because it can be used with patients with motor deficits. I forgot the animation again, sorry. And so first, it's interesting because it can be used with patients with motor deficits, but also it's interesting because of its neurofunctionally equivalence to motor execution. So as you can see on those fMRI images on the left that depict in dark blue at the bottom line here, the shared activations between motor imagery and motor execution, you can see that over the sensorimotor cortex, you have a similar activation as well as around the visual cortex, the occipital areas. So on the EEG on the right here, you can see that motor imagery is associated with an ERD, event-related desynchronization, during the task, followed by an ERS after the task stopped, just like it is the case for motor executions, though at a lesser extent. It should be noted, however, that those modulations deeply depend on the way motor imagery is performed. So basically, it's mostly what we call a kinesthetic motor imagery, that you can see here, which consists in feeling all the sensations related to movement, which is associated with modulations of sensorimotor rhythms. On the other hand, if you practice rather visual imagery, that consists in simply visualizing ourselves or somebody else performing a movement, you will mostly activate or generate modulations of activities in the visual cortex. So this is very important to remember, that motor imagery triggers different neurophysiological patterns depending on the way it's performed. So if we want to use motor imagery in order to modulate sensorimotor rhythms, be it for neurofeedback training or to control a device, we should train our subjects to perform kinesthetic motor imagery, which is not an easy task, especially because we have no feedback on what happens in our brain activity while we perform motor imagery. So we cannot be sure that the strategy we're using is really efficient. So optimizing this cognitive training is a challenge per se. So to summarize, BCI-based neurofeedback can be used to investigate neurophysiological mechanisms, for instance, better understanding the role of beta or sensorimotor rhythms on motricity. But for it to be possible, subjects should first be trained to self-regulate neuromarkers of interest. So for instance, doing kinesthetic motor imagery to modulate sensorimotor rhythms, knowing that this self-regulation can also be used as a cognitive training to improve or restore, for instance, motor abilities, and also for controlling external devices using BCIs. And to facilitate this training, subjects should be provided with a feedback that enables them to be aware of their brain activity modulations, knowing that this awareness can itself, and this is what is written here, can itself be used as a means to guide and motivate end users during their cognitive training. So in the second part of my talk now, I will present some of the work we have been doing the last years on each of those axes, starting with the investigation of mechanisms. So we were talking earlier about the mysteries that remain regarding the functional role of sensorimotor rhythms and of beta activity in particular. So beta has mostly been suggested in the literature to underlie motor vigor. So larger beta ERD would reflect or enable a stronger movement. But some experiments show somehow reverse patterns. And so some authors now suggest that beta would not or would rather reflect motor adaptation or flexibility rather than vigor. So in order to disentangle this, we led 2 experiments on 30 healthy participants each. So each experiment consisted in using EEG-BCI-based bidirectional neurofeedback in order to enable up or down regulation of beta activity before performing a motor task. So in the first experiment here, participants had to squeeze a dynamometer as strong as possible. So with this task, we wanted to investigate whether beta ERD correlated with motor vigor. In the second experiment now, participants had to do a tapping task. So they were asked to tap as fast as possible or as slow as possible. And so in this second experiment, the metric for motor performance, depending on the instruction, so it was the speed, but either they had to do as fast as possible or as slow as possible. And so following the flexibility hypothesis, a larger beta ERD in the second experiment would lead to faster movements when instructed to move fast and to slower movements when instructed to move slowly. So for both experiments, each trial would consist first in a fixation cross, followed by a neurofeedback phase. And when the beta regulation performance was maintained above a specific threshold for 2 seconds, participants was asked to press the dynamometer and then to report how helpful the task was, or to do the tapping task. So in the beta down condition, participants were invited to perform kinesthetic motor imagery to reduce their beta. In the beta up, they were asked to do relaxation, and in the sham condition, they were asked to stay still. So results now. First, they showed that the paradigm globally worked as participants elicited a larger ERD here in blue in the beta down condition as compared to the beta up condition. Nonetheless, there was a strong between subject variability, so that you can see here trial-wise. And those two graphs show per experiments the participants who were considered responders, so who would down-regulate in the condition down, up-regulate in the condition up, and yeah, that's it. And you can see that globally, on average, 60% of the participants were qualified as responders, so globally managed to modulate the beta activity in the direction of the neurofeedback instruction. So this is not surprising that it's so low if you know the BCI neurofeedback literature. We have around 40% of non-responders. What I would like to stress here is that among those non-responders, there were actually 13 to 21% who did not respond at all. They are in gray. But also 18 to 30% who actually modulated their brain activity in the opposite direction, who down-regulated in the condition up and up-regulated in the condition down. And this distinction between anti-responders and non-responders is most of the time not reported in BCIs and neurofeedback studies. So most of the time we just compare responders to non-responders, and I have the feeling, and you will see afterwards why, that it might be an issue because it makes a huge difference on the outcomes. Oh, it's automatically moving now. There is a huge difference on the outcome you can expect depending on if the person doesn't regulate at all or if they self-regulate in the opposite direction. So I will come back to that. So anyway, given this variability, Emmeline, who is the postdoc, the brilliant postdoc working on this project, she performed mixed effect models, taking the participants as a random factor. So in a nutshell, results show that for the first experiment, on grand average, we have a significant effect of pre-movement beta power on the force exerted. So as you can see here, so the three lines are the three conditions, but whatever, it doesn't matter because we really look subject-wise here. And you can see that basically the lower the beta, the higher the force exerted. And so this result seems to confirm the hypothesis of beta coding for motor vigor. Now, if we have a look at the second experiment, we can see a significant interaction effect between beta power and speed instruction on movement speed. So this is logical, of course. And what is very interesting that we have a lower beta power associated with slower movement in the slow condition and with faster movement in the fast condition. So basically, here you can see the beta power depending on the speed, and you can see that higher speed is associated with lower beta power and in the fast condition, and lower speed is associated with a lower beta power in the slow condition. So this suggests that actually beta would actually code for flexibility, and this could have a major impact basically on the way we see motor symptoms in Parkinson's disease, for instance. So the issue would not only be bradykinesia, so slowness of movement, but rather flexibility issues that could be it could be flexibility issues that are reflected by bradykinesias or other symptoms in patients with Parkinson's disease. So that was the first part. So now I would like to talk to you about another project on the neurofeedback for brain activity awareness. So for this second project, which is led by Mathieu and Antoine here, I will take the example of training athletes. So as you may know, if you're an athlete yourself or musician, motor imagery is used a lot in sports, be it for training problem-solving abilities, for improving self-confidence, or regarding more kinesthetic motor imagery for the optimization of movements and for functional rehabilitation. A lot of studies have been led to assess the potential gains following motor imagery training. So I will not elaborate here, but for instance, I will show just 3 of them. Here you can see a study where they did some weightlifting. So you have pre-test, post-test for a group, a control group, and a motor imagery group. And you can see that the motor imagery group increases more, significantly more than the other group in a post-test. And this is another example where you can see the time required to perform a kata in judo, depending on the group, in pre-test in white versus post-test in black. And there are 3 groups. So one control group who doesn't do any motor imagery, another control group doing only slow motor imagery, which means they had to imagine the kata slower than what they do usually do. And the fast motor imagery group, they had to imagine it quicker. And you can see that in post-test, the fast motor imagery group increases the speed significantly, while the slow motor imagery group decreases the speed significantly. So there's an impact of motor imagery also on the time execution. And this is the last example with music playing. So there were novices who had to learn to play at piano. And there were 3 groups, one with physical practice only, one with mental practice only, and one with control group. You can see here the number of errors depending on the training session day. And you can see that the physical practice group increases or decreases the number of errors, so increases performance quite quick. And you can see that motor imagery group takes more time to reach the same performance, but still they reach the same performance after some time, while the control group doesn't improve at all. So what is important here is that those studies show that motor imagery can be used to improve strength, precision of movement, speed of movement. And globally, this is if this topic is of interest for you, because I think that as BCI researchers, we should really read more the literature on sports science, because there have been a lot in sports science on this topic. And there is a very good researcher who happens to be French, who's called Émeric Guillot, who's written a lot of papers on that. So he's written books in French, so if you want to learn French, you can use this book, but otherwise there are also scientific papers. So the main issue for people who want to practice motor imagery in sports is that they do not have any means of objectifying and of quantifying performance and progression, because they don't have any feedback during motor imagery training. And this is where BCIs and neurofeedback come into play, because providing neurofeedback enables athletes and their staff to receive indications regarding the neurophysiological activity modulations during mental training. And if you remember what I said earlier, it happens that depending on the way you perform motor imagery, so be it kinesthetic or visual, there will be different outcomes, and because there will be different activations or different patterns. So what is of interest for them if they want to improve their motor abilities would be to do kinesthetic motor imagery to modulate sensorimotor rhythms. And so the neurofeedback can be used to do that, so that they can objectify, quantify, guide, and motivate the athletes. And so what we've done is that we've created software with a user-centered approach, because the risk, as you may know, is that we do something, if we don't apply a user-centered approach, it might happen that actually we develop a tool in the lab, and when it comes to the field, it doesn't meet the needs of the athletes or the end users, and so they never use it. So what we did is that we gathered a group of stakeholders, so athletes, coaches, physiotherapists, etc. And we discussed with them, designed the first software that you will see here, provided it to them. So there are several Olympic and Paralympic athletes whose used it for preparing the Paris Games last year. And so the next step now is to collect the data and gather again our group to get some feedback and reflect them on the next developments. So you can see that if I manage to start, I have to. Yeah. So here you will see that basically we use two types of feedback, what we call an evaluative and explanatory feedback. So during motor imagery, the athletes, so here they imagine a movement, and it's already finished. Oh yeah, they have this simplified version of the brain showing the activation in the motor cortex or visual cortex, of course in a simplified version. And here you have in the middle this target that is, let's say, an FPS target like in video games. So the better you are, the smaller the target is. And so we've been using this with different athletes. So here you can see an example from yesterday with, if some of you like swimming, you may recognize Summer McIntosh. He's Olympic Canadian Olympic champion. So we've been using this software with them, and we are currently working together to improve it. Okay, so let's continue. So basically, we are convinced here that, why I didn't explain really, we use this explanatory feedback, especially for novices, because we are convinced that when you're a novice and you practice motor imagery, you need to build a mental model of what is motor imagery and what are the consequences of motor imagery on your brain activity. So this explanatory feedback enables athletes to check when they use this or that strategy, which activities are happen in their brain. But then when they become more confident and more expert, they don't need this representation anymore. So it will be more the evaluative feedback that will be useful. And so they need a more precise evaluation of their performance. And so this is why we would switch towards evaluative feedback. So more globally, I would say that the form and the content of the feedback that we provide to people who want to use, learn to use a BCI has to be adapted to their profile, which leads me to the third axis here on how to optimize globally the neurofeedback training procedures to optimize self-regulation skill acquisition. And here we'll take the example of the project of Elise Grevet on stroke rehabilitation. So here you can see a medical doctor testing a patient. So basically, after a stroke, many patients have motor after effect, as you all know, I guess, and often hemiparesia or hemiplegia. And standard, let's say, rehabilitation technique would consist in asking the patient to try to move or to imagine moving in order to trigger sensorimotorism and the activation of the sensorimotor cortex, and at the same time to mobilize the arm in order to close somehow the sensorimotor loop. The problem here is that when the doctor mobilizes the arm, he doesn't know exactly when the patient is trying to move. So this sensorimotor loop cannot be really closed because the feedback is often not synchronized with the intention of movements. And so this is here, once again, that BCIs and neurofeedback are very interesting because you can detect the ERD in the sensorimotor cortex and then provide a synchronized feedback. So this here our hypothesis. Oh, sorry, I had this slide I forgot about. So it's just an illustration of what I just said. So you can see here the activation of the sensorimotor feedback, and then you can provide the sensorimotor cortex, and you can provide a sensory feedback. So here I have two examples. I have an arm moving on screen depending on the amplitude of the desynchronization. And another example from EPFL where they have functional electrical stimulation. Okay, so this you all know, I think, that it's we kind of have a lot of scientific evidence that it is useful to improve functional rehabilitation after stroke, and yet it's not used really in clinical settings. So our hypothesis is that actually the efficiency would not be enough, and that you need to improve as well the acceptability of the system. And to come back to our user-centered approach, we argue that clinical efficiency or BCI controllabilities could greatly benefit from the maximization of agency and of acceptability. So this comes from a model that we proposed some years ago. Yeah, and this model was saying that BCI efficiency is also determined by non-specific factors, including the feeling of being in control, which is what we call agency here, and also the relationship with the technology. So if you're anxious, basically, to interact with the BCI, you will not engage in the task, and you will have less attentional abilities, and so less good performances. So here, I will present very briefly the work of Claire and Elise on those two aspects. So first, with Claire, we led a study on 25 participants who were asked to self-regulate their sensorimotor rhythms through motor imagery of their dominant hand using different feedback with increasing levels of transparency. So our idea is that the more transparent the feedback, the more agent you will feel. And what do we mean by transparent? It means the more consistent the feedback is with the task, the more agent you will feel. So our less transparent feedback was this pendulum. So the amplitude of balancing of the pendulum would depend on the amplitude of the ERD. The second, more transparent feedback was this hand moving on screen, as the one I showed before. So the larger the ARD, the larger the movement. And then we had a third condition that was supposed to be the most transparent, with this hand moving on screen, plus some motors here on the wrist, and yeah, it's the wrist, and providing motion illusion. So the same as earlier, we had trials where healthy participants had to imagine moving for some time, and then depending on the ERD, they would receive this continuous visual feedback. And so here are the results. First, on the left, you can see the neurofeedback performance depending on the condition. And you can see here that the best performance is obtained with the motor imagery on the hand, the virtual hand only. Basically, I will just briefly mention the hand plus the vibrations. The vibrations were not well synchronized with the imagination, so it really deteriorates the performances. Anyway, what is important is here that we have the best performances with the virtual hand only. And what is interesting is that actually, when you look at the feeling of agency that has been reported, is also with this virtual hand only that it was considered or ranked the best. And here, this graph represents the correlation between the level of agency and neurofeedback performance. And you can see that there is a positive correlation. So the question here was, is it the feedback that itself, the transparency of the feedback itself that influences neurofeedback performance, or is it agency? So we did some mediation analysis, and we saw that basically the effect of feedback on neurofeedback performance is mediated, almost totally mediated by the agency. So which means that agency has a major impact on neurofeedback performance. And so what we did afterwards is that we tried to investigate a little bit further those correlates of agency. And here you can see the correlation for each electrode, each frequency between the EEG power and your feedback performance. And of course, we have in blue here the negative correlation between EEG power and neurofeedback performance, because neurofeedback performance is actually the size of the ERD, the beta ERD. So this is logical. But what is interesting here is that in addition, we have this pattern in theta with a positive correlation, meaning that this frontal medial theta positively correlates with neurofeedback performance. And here we come to my favorite graph, and you can see the graph representing neurofeedback performance correlation with frontal-central theta. And what we did, or what Claire did, is that she separated between low scores of agency, medium and high scores of agency. And what we can notice here is that when people feel agent, so between agency between 10 and 8 and 10, this frontal medial theta doesn't really correlate with neurofeedback performance. It's only when they feel not agent, so that when there is a lack of feeling of agency, that there is a big correlation, which is also very consistent with the fact that this frontal medial theta would actually reflect some conflict monitoring processes. So the second study, very quickly, because I have to wrap up now, is about acceptability. So this is the work of Elise. So the idea here is that acceptability, which corresponds to the intention to use the technology that is basically modulated by the perceived ease of use and the perceived usefulness of the technology, is not at all studies in the field. So we've had a review of the literature, and basically there are 3 or 4 papers in the whole BCI literature about this aspect. So what we did is that we created a model based on different existing models in other fields, saying that this acceptability can be determined by characteristics of the system, like the scientific relevance of the system, social influence, individual characteristics such as anxiety, self-efficacy, and facilitating conditions such as agency or playfulness of the system. And what we did is that we validated this model and assessed it in the population, general population, with more than 700 persons, representative of the population, and with 140 patients. And we looked at the most influential factors for each of these samples. And results show that for the general population, the perceived ease of use is mainly influenced by the playfulness of the system, and that this perceived ease of use only influences a little bit the perceived usefulness, that is mainly influenced by the relevance, the scientific relevance of the system. And finally, the intention to use is mainly influenced by the scientific relevance and the benefits on risk ratio. So for the general population, if you want to convince people to use the system, it has to be scientifically relevant and have a low benefits on risk ratio. Now, when it comes to patients, we don't have the exact same patterns. So again, the perceived ease of use is influenced mainly by the playfulness. But this perceived ease of use has no impact at all on perceived usefulness and intention to use. Rather, the perceived usefulness will be once again influenced by the scientific relevance, but also, this is important, by the level of anxiety, autonomy, and self-efficacy of the patients. And finally, the intention to use will be, of course, influenced by the scientific relevance, but also mainly by the profile of the patient, their anxiety, their autonomy, and their self-efficacy. And hence the importance of really adapting the systems, the procedure, the rehabilitation procedures, for instance, to reduce anxiety and increase self-efficacy so that people feel like they want to use the system, and so that it's efficient. So to conclude, I would say that we are lucky to be in a field with a lot of public and private investments and opportunities, but at the same time, we have the responsibility to answer high expectations from investors, of course, but also and especially great hope from end users. And the only way I think that we can answer this hope is to use user-centered approaches to be sure that we meet the needs and expectation of the end users. And with this, I would like to thank you very much, and thank you all the people I'm working with. They are fantastic. Thank you very much.
Thank you very much, Camille, for your presentation. Are there any questions from the public? We have one clap, but not one hand up yet. Okay, I can warm up. I was wondering, so in I guess the one of the last things you were talking about agency, and there was this you're talking about the correlations between, I think it was agency and the effectiveness of the neurofeedback.
Yes.
Did you actually look at did you do any causal analysis and all of that, or was it all correlational for now?
Yes. So we did some there. Okay. So here we did some mediation analysis with so they are the best which is the best to my knowledge we can do for neurofeedback and well, the feedback, agency, and performance. But for this one, we don't have. For me, based on our protocol, we cannot really extract any causality link between both. So it's a real question that we have, and we discuss in the paper, by the way, of the fact that is this modulation of theta a consequence of the performance, or is it something that precedes the performance? So there's a lot of literature on the sense of agency and on the neurophysiological correlates of agency, and more specifically on the feeling of agency and the judgment of agency. So the feeling comes before and the judgment after you've perceived the feedback. But in our case, it's really difficult because, you know, it's we have this continuous visual feedback. So basically, if we look sample by sample, we cannot really say if the person is judging the feedback they've received before, or if they're field agent or not for the coming trial or the rest of the trial. So this is really something we would like to investigate further in the future, but which would require a different protocol. I hope it answers your question.
Yeah, it does. This is the one I was talking about, the other one, yeah. Okay, Sinis, you have a question.
All right, thank you. So it's a question that I always wanted to ask you. I believe I never did. It's not really on the results you showed today, but some older things you were doing. So I remember when I was writing my thesis, I was influenced a lot by this paper flows of the motor imagery training protocols. And what I always found particularly interesting, but also very confusing, is the part about instructional design. So the argument there was that the protocols that we normally train people with for motor imagery, they just don't tell them how to do the motor imagery, and especially how to do it in a kinesthetic way. And I was always finding that this is very true, but in the same time, how can you make a protocol with a good instructional design? So can you elaborate a little bit on that?
Yes, this is a very good question. It's been torturing me for some time as well. So basically, so this paper on the flows of current protocols, it's a paper from Fabien Lotte, 2013, I think. And so basically, the thing is that in BCIs, sorry, it's been really annoying. I think there is something, sorry, I will just stop the sharing. So basically, most of the time in BCIs at the moment, what we do is that we ask people to do some calibration, and based on that calibration, we train machine learning algorithms, and those machine learning algorithms will basically try to figure out which are the most separable patterns for both tasks, or how many tasks you've got, and would then train people to replicate those patterns in the future. But the problem, I think, if we do that, is that we don't have an idea of how to instruct people to replicate some patterns that we don't know what they mean. And so this is why if the starting point is the use of machine learning from the start for motor imagery training, there is no answer to your question, I think, because you cannot teach people if you don't know yourself what are the patterns that have been discriminated. So this is why I would argue that maybe a better solution, and this is what we've been working on together, would be first to have a more pure neurofeedback approach where you simply show people their brain activity, or simplified version of their brain activity, why they perform motor imagery. And in that case, you reverse the problem. So you teach them, you give them instructions based on sports science. They've got a lot of insights on how to teach people to do kinesthetic motor imagery. Most of the time they do progressive motor imagery. So they, for instance, in a free throw in basketball, they would do the free throw first with the ball in their hand, and then they would reduce the amplitude of movement. And so they do that. And with this, they see that people, by conditioning, they can learn to see how to activate their brain, their sensorimotor cortex. And once people can do that, then you can go to more machine learning and train your algorithms, this time on patterns that you know how to instruct people to modulate them. So I would say that it's really dependent on the approach you used. And indeed, if you use machine learning from the start, I don't see how you can apply instructional design. I hope it answers your question.
Yeah, no, it's clear. Yeah, because I thought we need somehow to tell them how to do it. But essentially what you're saying makes more sense. It is try stuff, see first with neurofeedback what seems to do something in the signals, and then use that, which makes sense.
Yes.
I think both me and Rainy, we have been doing stuff along these lines. By the way, the paper was 2013, I think.
Yes.
You expected her to remember the year. Okay, the last final question before Camille has to go pick up her child.
I think my husband is back from work, so he can
Okay.
he can handle the baby. So it's
Okay. So we will have time for an extra one more question, if there's any.
Maybe not a question, just a thank you. I was only half focused. Very interesting work, but I will follow up with you at some point. We anyway need, no? I did not ghost you all the emails. No, thank you so much for giving your presentation. And there's a lot of things which might be overlapped, no? In terms of research or what we could be doing, as Siemens was mentioning.
Yes.
Anyway, I have to run to my next meeting in a few minutes. So
Okay.
the ones ask questions.
I'll just have another quick one. So I think the research you were presenting today, it seemed to be all based on one session of training, or was it more longitudinal?
Okay, so not all of it. So the first aspect on the investigation of the mechanisms, the work from Emmeline on motor vigor and flexibility, this one based on 1 or 2 sessions depending on the studies, because it was not at all about learning. It was really about straightforward effects. Then with the athletes, it's a continuous process. So they use it as long as they want. And this is really a translational project with no, it's not a very standard protocol. The idea is really to be on the field in ecological conditions and see how they can use the system. And actually one of our metrics is how long they use it and for how long and how often they use it. And lastly, for the third session, we are actually on, for the third project on stroke rehabilitation, we are really actually currently undergoing a clinical trial with 50 patients. So we are currently almost half of them, so 20-something patients, and 15 sessions per patient. So this is a huge, a rather huge clinical trial that we're currently undergoing. And this is really something important. And we were talking about that because we are involved together on a European consortium with Simis and Sebastian and Rainy. And so it's really something that is very important that we have time and we take the time to do several sessions, because we cannot expect really learning if we only do 1 or 2 sessions. So this is something really trying to fight for. But the problem is that at the moment we don't have the data to tell people how many sessions they would need to be able to reach control, and there's a lot of variability, and so we need more data to understand this variability and to be able to adapt our protocols to ensure that we can reach some learning.
I guess there's two. So I was going to ask about that, so how long until they learn, but also how frequently do they have to train? I'm guessing that's also part of the.
Yeah, to the best of my knowledge. Yeah, to the best of my knowledge, there's no evidence based on neurofeedback or BCI data, because back in the 90s there would be people like Andrea Kubler who would do experiments like on 20 or 200 sessions or something like that. It doesn't exist anymore, and at the time they would do it on 1 or 2 patients. So we would need many sessions for many patients or many participants, and we don't have that at the moment. So what we can do is just looking at other fields and at like psychology or ergonomics. And what I've seen so far is that they would advise 1 session every other day, so once every 2 days. And like 3-4 sessions per week, and for a long time. But then there is so much variability, and it also depends on the protocol, so you cannot really say exactly.
And then you also have the anti-responders and the non-responders.
Exactly.
And the whole thing just becomes exponentially more complicated.
Yeah, exactly.
Okay, well, last chance for any last question. If not, we'll just thank our speaker. Thank you very much. It was very good.
Thank you very much for the invitation.
Thank you for accepting.
For attending.